The Origin of Species by Brian Morris. When looking at plants and animals, one notices characteristics specific to sets of individuals that set them apart from other individuals. Organisms can be grouped into clusters based on phenotypic similarities and defined as being separate from other organisms and clusters of organisms based on these differences. For example, there are numerous individuals of cats that are clearly like but distinctly different from the many individuals classified as elephants. These clusters of similar and distinct organisms are what we call a species. Instead of a single type of life evolved from a distant ancestor, we see many species, very different from each other, with numerous varying adaptations. Natural selection explains adaptations within a species, but how do species arise independent from each other? Speciation is a discontinuity from a single line of evolving individuals. So the question is, how does speciation fit into evolution? And does evolution allow for and explain speciation? Defining exactly what a species is no easy task. Sometimes it would seem almost arbitrary, or up to one's opinion, where one species ends and another begins, because some of them can look so similar to each other. We can clearly tell that a cat and an elephant are different organisms, but boundaries between species are not always so clear. The finches described by Darwin on the Galapagos all look nearly identical to each other, but are classified as being different species. Before discussing how a new species comes to be, we first need a way to define a species as being separate from others. To answer the question, what is a species, biologists have come up with dozens of different ways to define a species. All of them rely on some type of mechanism. This may be necessary, as what a species is could simply be a label created by humans to help organize the natural world. In 1942, Ernst Mayr and colleague came up with the biological species concept. The biological species concept is based on reproduction. Individuals of a species are of the same species because they can reproduce with each other, but not with individuals of another species. Species are defined by reproductive barriers, by whether or not they can mate and produce viable offspring. This definition can be tricky though. Sometimes two different species can reproduce and form a hybrid. There are two more concepts worth mentioning. The phylogenetic species concept, which is about defining a species as an irreducible cluster of organisms with an ancestral line different from other clusters and diagnosed by their differences. And there is also the evolutionary species concept, which defines species through an evolutionary history of ancestor to descendant progression. This is useful to paleontologists who cannot study them live. Speciation is lineage separation and bifurcation. It requires barriers to gene flow so that two populations can evolve independently from each other. If gene flow between them was unrestricted, the gene pool would just homogenize and undo any speciation. Here we will discuss how speciation happens, starting with allopatric speciation. Allopatric speciation happens when a population is split into two parts by a geographic barrier like a mountain or a river, or if a species is dispersed along oceanic islands. This is called vicariance, and in time, the two halves of the population will drift, mutate, or through selection, speciate. The point of vicariance is a reproductive barrier has been established between the two populations, halting gene flow. If the two gene pools are sharing alleles, they will homogenize and not be able to split into sister species. For example, a large population of rabbit are living in a forest. Climate changes and flash floods divert a major river to run through the middle of the rabbit population, and the two halves of the populations can no longer intermingle. Over a long period of time, mutation and drift can cause the two separated gene pools of the rabbit populations to become different enough that they are no longer able to reproduce with each other. Should the two populations be reintroduced, they may not be able to produce viable offspring anymore. When that point is reached, the two populations of rabbit will have said to be speciated by vicariance. Alternatively, in a way called peripatric speciation, a small population may break off of the main population of a species and move off to inhabit a new area. In time, if gene flow is absent, the migrant population can evolve away from the original population. Should the migrant population eventually return, they may not be able to reproduce with the original main population and will now be a new species. This is called peripatric speciation. An example of peripatric speciation would be if a small population of rabbits were to break away from the main and move to take advantage of a new food source in a different type of habitat. In time, the migrant population may change, and should they return to the original population, they could be considered a new species. This requires, once again, restricted gene flow between the populations and a lot of time. It is considered allopatric because the populations are isolated from each other and unable to share genes. In some cases, the returning species may outcompete and replace the original population. In the fossil record, this would look like one species instantaneously evolved into another, which is a phenomenon called punctuated equilibrium. Another mode of speciation is called peripatric speciation. It requires limited gene flow between two adjacent populations and often happens on the borders of two ecological zones. Two populations of a species, one in a tropical area and another in the savanna, whose territory overlaps at the changing point, may experience reduced gene flow if they do not cross habitats to mate. With enough time and lack of gene flow, the populations can diverge. Eventually, even with limited gene flow at the beginning, the two populations may develop different mating preferences. Perhaps certain rituals of mating or calls are no longer recognized, and gene flow between the two populations finally ceases. Should they still occasionally interbreed later in time, they may produce hybrid offspring. The hybrids they produce can be infertile or less fit than the parent species, and in both cases they will fail. Lastly, there is sympatric speciation. This happens within one population when a new species arises from within a single gene pool. Here there are no obvious barriers to gene flow like mountains or rivers, and this method is much less common than the other methods of speciation, allopatric and peripatric. This can happen through sexual selection. If mating preferences cut off a group within the main population, perhaps a particular pheromone is desired by some and undesirable to others, or mutation of coloring is appealing to some and not others, or even changing in mating calls too, can cause some individuals to be unrecognized by the main population. Or it is also possible that some alleles are selected for so strongly that gene flow cannot homogenize the divergence. One more method of sympatric speciation is polyploidization. This is most often seen in plants. As a matter of fact, many angiosperms, or flowering plants, came to be through polyploidization, because it often requires one to mate with oneself. Plants sometimes having both male and female parts can work this way. Polyploidy happens when the chromosome number doubles through mutation or non-disjunction. Because of a mismatch in chromosome numbers, it can no longer reproduce anybody but itself in this case. Sometimes, the chromosomes of two different species can mix, creating a hybrid with a different chromosome count from either of the parent species. This is called instant speciation because a reproductive barrier is immediately established between the hybrid and the parent species. Adaptive radiation can describe the formation of many new species as well. If a large number of niches in the environment suddenly open up, a species can rapidly spread to take advantage of these new resources, and, in time, diverge into many new different species, like how the mammals exploded after the extinction of the dinosaurs. Speciation is a slow process in sexually reproducing lineages, requiring hundreds of thousands, if not millions of years to take place. It also requires barriers to reproduction, otherwise gene flow will thwart any divergence. It is important to note that species do not evolve for the betterment of nature and the environment, nor do they evolve out of some requirement to fill an ecological niche. New species are the result of the splintering of lineages, a random process of divergence from ancestor to descendant.